All right, let's come back. Start from where we started. Okay, so we briefly talk about solid platform and the Illumina platform. So just to keep in mind, the major difference between the two technologies are, number one, their amplification procedures are different. For the Illumina, it's bridge amplification. For the solid, it's emotion PCR. And for the detection method, it's different. For the Illumina, it's synthesis, sequencing by synthesis. So at the time that you put more nucleotides on, you do the sequence. And for the Illumina, for the solid, it's sequence by ligation. And one other major difference between the two platforms are their data structure. And for the Illumina, it's more like a base space. That's what we call it. It's you get ACGT and direct sequencing. And for the solid platform, you got color space. And it becomes really confusing for the color space. But it has a lot of good features that built in. Okay. And I'm going to briefly go over several other platforms, but it will be very brief, not go into any detail. So the first one is the 454 technology. So what they use is something called the parasequencing. And to a certain extent, if you still think of amplification and sequencing, okay, this is something that you want to distinguish. You can see here is for the amplification part, it's very similar to solid technology. You still got the emotion PCR and put those DNA molecules around the beads. Okay? And for the sequencing part, they use something like called a parasequencing. I'm not going to go detail about that. And what happens is once you put in one nucleotide in, and you will detect some signals, and these nucleotides, when it hybridizes, it will release something called a paraphosphate, and then do some reactions and excite the color, so you can get the image of it. So again, the point, I'm not trying to get into detail of this about the biochemistry part, but for the 454 technology, there is really good features of that. Is the length of the rays are really, really long. And for the Illumina technology, it goes to 100 base pair, and for solid, it's 75 base pair. But for 454, it's a 400 base pair so far. And as we mentioned that this 454 has been purchased by Roche, which headquarters here. So they have a very nice poster in their company. And I wasn't able to find it online, so I make my own. So the point here is once you get a nice picture here and you want to play puzzle games, right? There's two choices. You can play this puzzle game, so we break it into 4 different pieces, and each one is pretty big, or 200 ones, and each one is very small, right? And which one you want to play? That's the question. And of course, if you want to torture yourself, of course, this is the way to go. But when we do the sequencing, especially when we do assemble the genome, once we got a short reads and we want to assemble the genome, and this is a much easier way to work with. So that's the point of their poster. I thought it's very neat. Okay, so yes, they call it length matters. So not only the number of rays you will be detecting. As I mentioned in the 454 technology, the throughput is way lower than the other two competitors. And when you translate to the cost, it's pretty expensive. So it's kind of the problem. All right. And next technology I want to go through very briefly is Pacific Bioscience technology. So this is one technology that you heard a lot of press. They raised a lot of private money and to develop their company. And it's very successful. And they have really their advantages. They do have their troubles as well. So their major advantage is the single molecule real-time machine. So it doesn't have the amplification procedure. You don't need to do the amplification part, which anything when you do PCR, you have the opportunity to introduce the noise, and which you don't have to do it. So what happens is for each chip, so you can see here is each chip with a waveguide 100 nanometer hole. So basically, you can watch the DNA are synthesized. So for each individual hole here, it's about 100 nanometer, and then they have a polymerase really sitting inside the hole. Okay? Once you got the DNA molecule going through it, okay, and you have this extraction from the bottom, and then, of course, those has to be color-labeled nucleotides, and then it will show up different colors. And so you can see here. So this is the double helix, the DNA here, and this polymerase. And you can see if this is the yellow one supposed to hybridize to extend it to here. And what happens is once it is put on the DNA, and at this time you give excitation, and then you will observe this yellow signal at this particular time points. Okay, and then when you do another one, and of course you release it. When you do another one, and next one is a blue one, and at this point you will detect a blue signal here. So it's a kind of you're watching a movie, and the polymerase is sitting inside the hole and grab a sequence and just go through it. And every time it shines a different color, so you will know the sequence of that particular molecule. It's a very smart technology. And the most important part of this is really long rates. Okay, it's really long rates. It's 1,000 to 1,200 base pair, and about 5% of rates can go to as much as 5 kilobase. So you can see this can be a very important application, and become assembly is much easier. And once you study RNA, you can really sequence the structures, the detailed structure of the RNA without going through those alignment hassles. Okay. And another thing I didn't put on here is the rumor is they can treat DNA methylation, the methylase they see, as a different color. So you can do that. So you don't really need to do, when you measure the DNA methylation, you don't really need to do any modification. Just go through it and it will work. Okay? So it's very good technology, very smart. But the problem is they don't have too much machine at this moment. For some reason, they only have dozens of machines deployed nationwide, and they better get those machines going. And another technology is called the helicose technology. And this is a technology, unfortunately, just delisted from the stock market. So they are not going to make it. Okay, and but this is the true single molecule sequence. It's the only true single molecule sequence. There's no amplification at all. It's very easy, simple steps. And of course, I heard about it. I never touched the data. And so you can, if you think of Illumina solid technology, when you go through those amplification procedures, either bridge amplification or emotion PCR, the library preparation cost is a very significant part of it. But for them, it seems that they only just put a poly T at the end of the DNA. That's the end of it. Done. So it's very smart technology, and unfortunately, they didn't make it. And they published a Nature Biotechnology paper in September 2009 in the single molecule sequence of an individual human genome. And their read length is from 24 to 70 base pairs, and they had 28 coverage, X coverage. So for a moment, this is the most affordable technology to sequence the genome, and it is killed by the market later on. Now, one of the problems is measuring a small amount of DNA is really, really difficult. A lot of people don't want to work on that. I mean, it's painful. And I don't understand this, but I heard that the alignment is tricky, but I haven't seen the real data, so I don't know why it's tricky. All right. But anyways, they're not going to make it. And this is the technology certainly made it, okay? And it will become a big splash in the next couple of years. It's called Aaron Torrent Personal Genome Machine, okay? And this is a company that was founded by the same founder of 454 technology, which is the first real high throughput sequencer. And they were just last year, I think, were purchased by Life Technology, the same company that has a Solid, with $350 million. At the time, they don't even have a product. And just the idea worth of that much. And now they certainly have the technology available, and actually we had two sitting in Harvard Edinburgh's lab. And the principle of this technology is when a nucleotide is incorporated into a strand of DNA by polymerase, a hydrogen ion will be released. So this is what the chip will look like. So it's a high-density array of wells. As you can see, there's so many small wells. Using the semiconductor technology, with each well holding a different DNA template. So basically, you put a DNA templates into it. Beneath the well is an iron-sensitive layer and a sensor. So it's not imaging-based thing. You detect the signals by the sensors. So when you sequentially flood the chip with one nucleotide after another, okay, if you flush into, for this well, you drop into an A, it's supposed to be the right one. And when it gets into this DNA molecule and being hybridized, put that on by the polymerase, and if there's a match, a hydrogen air is released, okay? And the change in the pH of the solution will be detected, okay? So think about this. This is not, so why those sequencers are so expensive, right? $500,000 and $700 million. And a lot of those are the very refined, very good cameras, high resolution cameras. Those are very, very expensive. For this technology, we don't need to see the image. We don't need those cameras. And this really dramatically brings the cost of the sequencer itself down, okay? It costs only $50,000 for each machine. So it's probably cheaper than some of the real-time PCR machines. And it's very affordable by small labs and also clinical labs. And it's very, very interesting technology. And also, there's another good thing of this is sequence is really, really fast. It's unlike the other two, so you have to prepare the library for a couple of days and then do a sequencing run for a couple of days. And you can get everything done in several hours. So you can still get very good throughput out of it. So this is one of they call the personal genome machine. And there's another similar platform I haven't talked about. It's also from Illumina, and it's called MySeq. So you remember their big one called HiSeq, right? And they also have MySeq, M-I-S-E-Q. And this is also the competitor with the Aaron Torrent technology. So they are aiming at smaller labs, smaller amplifications, sequences, but you need to get the data quicker, and the machine itself is much cheaper. So this is something that you need to keep an eye on. Yeah. The quality of the data, not good. So we haven't seen the real data yet, and so far they're still trying to I think it depends on how far you go for it, and some of the locations, the sequencer are 10% of error rate. We haven't seen the real data yet, but it does have a lot of interesting applications. For example, Howard purchased that in his lab. He already have the solid, right? And he purchased in his lab. One of the reasons is once you bring your sample in, how do I know it's good, right? Instead of sequence the entire thing for $10,000, let's do some titration runs using the smaller machines and to see the quality of your samples. And then, if it's good, go for the big ones, right? So there's a lot of interesting applications, but the major thing the market they're aiming is still the clinical laboratories that I think our department is trying to get a few as well. All right. I'm not going to go through all this. All right. So far, this is an incomplete list of all the next generation sequencing platforms, and it's already numbered to 36. And there's several different niches when you select a sequencer. Of course, the pricing, is important, and the read length and repeat speed and portability, all different things that needs to be considered. And there's a very interesting review article, and I encourage you to read it. It's a comparison of major platforms, and they did really, really great job. For the market share that we are having is, so this is survey is conducted by the CHI Professional Marketing Service, reported just published not long ago, and we can see that Illumina, 57% of the labs has Illumina. These numbers doesn't add up 100% because some of the survey people, they have multiple machines, right? So, but you can see that Illumina is still holding the biggest market share. And I guess the market speaks for itself. And as in most new technology, getting good data from a sequencer initially is not easy. Okay, so we know that these machines are available back in 2008, 2009, but how you can really get the data, that's a tricky issue, right? Yes, I have funding, I want to do some sequencing, and I will send my samples into some core facilities, right? And initially, a couple of years ago, when we sent the samples to OSU core facility, at the time that we don't have it here, and it's Illumina machines, and it sounds like it's straightforward. It's never straightforward. The lasers doesn't point to the right direction, and all those things will happen. And the initial quality of the data is not very good. But later, and as the technology develops, it's become really, really much better. And the backlogs of core facility initially was a big issue as well. So you send your samples in, and after 6 months, they send your data back. All right, that's the amount of backlog they do have. Now, probably, that's still a problem. So there's still a lot of core facilities, they have big backlogs. And the situation has been improved dramatically in the past years, and as the technology becomes more stable, and the cost is still prohibitive. I mean, I'm talking about to get the sequencer itself is still less than $1 million, and NIH funds many machines through their large equipment grants, and that's how we get our first solid sequencer from NIH equipment grants. The big genome centers have a substantial advantage in technology development, and the biggest one is the Beijing Genome Institute, and by the way, for those of you who are not Chinese, it's not in Beijing. It's confusing. It's in Shenzhen, but it's Beijing. So the day that Illumina released the high-seq machine, it's available. They purchased 120-something machines instantly, and I think it generate a big backlog for Illumina as well, so they cannot deliver machines for many other people as well. And their sequencing course is also very competitive, and people sometimes send their samples in. Okay, the throughput advance of this technology is really scary. So we are talking about 2 years ago, 3 years ago, fall of 2008, and at that time that the sequencer itself can cover the human genome twice in one single experiment. Okay, this is 3 years ago, and by the end of last year, it can cover the genome 100 times already. So this throughput advance is really, really great. And the cost per megabase of DNA sequencing dropped dramatically. This is fantastic. So this is a Morse law. So you can see this is the log scale. This is the Morse law. That's for the computer science development. They also follow this log kind of rate. But we can see that for the sequencer itself, it beats Morse law dramatically. Okay. And how to read this is in September 2001, if you sequence 1 megabase of DNA sequence, the cost is less than $10,000. Okay, today, not even today. Okay, half a year ago, and the cost of sequencing 1 megabase is 17 cents And this is also including the overhead and of the core facility, the administrative costs, and everything together. It's 17 cents per megabase of sequence that you can get. And this is a 10,000 times drop of the price during the past 10 years. It can be fantastic, right? And actually, the reagent cost of that part is for 1 megabase of sequence you got, the reagent cost for Illumina platform is 4 cents. Okay, that's the amount of data you can generate with limited funding. And so think about that. It's rapid decreasing cost. The Human Genome Project, 13 years and $3 billion. And in 2007, they did a Watson genome, and it cost $2 million. And for Illumina, now, today, for 8 days, it costs about $10,000. And this is really 10,000 times reduction in 5 years. And the claim is whether we can sequence a genome in 15 minutes for $1,000 in the years to come. And it becomes really real while you look at where this trend goes, right? Okay. And the number of publications indexed by PubMed, of course, it increased dramatically when it goes to involve the next generation sequencing studies. Okay, I hope I convince you that this is a powerful machine. And but the issue is how do we deal with it? I mean, we got a powerful machine. What we can use of it? Or is there any interesting biological applications? So some of you have seen this slide. So what we can do with these powerful sequencers, and the first application will be sequence the DNA. Okay, this is why this was initially developed. You can do the de novo sequencing, meaning you sequence the DNA which you don't know before, and it's not human, for example. And you can sequence, do the reference-based resequencing. So in our medical school, most application is here. So the point of that is to identify the genetic variations, including single nucleotide polymorphism, copy number variations, and the insertion deletions. And also you can sequence, do the metagenomic study, basically to identify who is there in a mixture of microbiology species. And you can sequence the RNA because of the throughput. So it's called RNA-seq, called transcriptome-wide sequencing, and you can also sequence the small RNAs and novel non-coding RNAs. And you can also study protein DNA or protein RNA interactions, and we will have a couple of lectures on that. So it's called ChIP-seq for transcription factor polymer, and then polymerase II, or CLIP-seq for RNA binding proteins. We can use this to study epigenetics, including DNA methylation, histone modification, nucleosome positioning, and chromosome looping. So all these, most of these things will be covered in the class. For the DNA sequencing, and there's different scopes. So the first one is whole genome sequencing, okay, for human studies. We know that in 2007, and actually the work was done in 2008, and published in 2008. And this is the first individual genome has been published. It's Watson genome using 454 technology, cost us $3 million, $2 million. And in 2010, and then the 1000 Genome Project published, and the goal of this project is to identify a deep catalog of human genetic variations. Okay, we are not satisfied with only the reference genome. We want to know the variations on each individual's DNA. And there's two 10K genome project also ongoing. One is to sequence 10K species, and they want to create a sequencing zoo. And another one is the 10K individuals, and it's most on the cancer genomes. And whole genome sequencing also can deal with the animal studies, and there are a lot of push towards that because a lot of animals, we have been using that as a good animal models for the human disease. And for example, for the mouse genome and rat genome, different strains of animal models, and they want to really sequence them to tell me what is the genetic variation between different mouse strains, for example. And some are institute is leading that effort currently. Okay. Whole genome sequencing. Okay, many disease genomes will be sequenced in the coming years, and that's apparently true. And so one key thing that we want to identify from these sequences are the sequence mutations for the disease samples. So we probably know the single nucleotide variations pretty well, single nucleotide polymorphism, the common variants pretty well. But for different diseases, they have disease-specific sequence mutations, and how we can identify those is a big issue. And that's something that in the later lectures, I'm going to talk about the difference between SNPs and mutations. There are subtle differences. And another thing is to search for the structural variations. And these are the things that was not extensively studied due to the limitations of technology. Okay, and so we can get into that a little bit. And this is very important. So both germline and somatic variations may be of interest depending on the disease. For example, in cancer, we know that in the tumor samples, the DNA has been really messed up. There's a lot of bad things, especially structural variations, happened in the tumor tissues, and they are not in the blood DNA. So it's not in the germlines. So there's a strong push on the sequencing the DNA in the tumor samples. We call it somatic variations. Can be very important. But depends on what who you are. Whole genome sequencing is still very expensive. So far, the cost is about $10,000 per genome if you really want to get it in pretty high quality. And I think, okay, I have $10,000 and let's sequence one individual. But it turns out to sequence one individual doesn't really help you too much, right? And you need to have something to compare to. And we know that for some genetic studies like GWAS, you need hundreds of thousands of individuals to be sequenced or genotyped to get a real biological meaning. And sequencing 1,000 people is still very expensive, right? So far we only have 1,000 genome sequencing was done, and it has been a big push. So whole genome sequencing for many disease applications are not really possible. So therefore, targeted sequencing or transcriptome sequencing may be a substitute. It's a good thing to go, okay? And a lot of people think cancer is the genome of the cancer is really maxed up, and we think so too. And so you can see this cartoon. It said the angel said, God, the human genome code has been unraveled, and what God needs to do is just change the password, and you will not be able to understand whatever you are, right? And actually, cancer are pretty smart. Okay, they can generate novel mutations, novel variants. And you think you find out the markers, but that's not the marker, and they can really make rapid changes. And so there's a high rate of abnormalities, sometimes 10K mutations per cancer. And another issue is, yes, you got a couple people you want to sequence, and you sequence it, you find out variations, but the problem is only a small subset of those variations are driver mutations. Meaning those are the mutations that are the password for the cancer, right? And most of them are only passenger mutations that it's not having any biological meaning. And but how can we identify those passwords? How we decode that is a big issue. So question is it possible to identify the drivers in this tumor genome that drive the cancer progression? And is that possible to have the therapeutic selection based on genomics? So I will introduce one study to give you a hint what that really means. Okay, so ideally, so we talk about identify the variations that really causing the disease, the sequence to identify causing variations. So if you have unlimited resources, and of course we want to do the whole genome sequencing, which can help us to identify the variants in both non-coding regions and encoding regions. Okay, But since we don't have unlimited resources and we have to stay focused on, and it's possible that we can only sequence the exon part, most likely the money is there, okay, although not for sure. And also doing transcriptome sequencing and to identify variations on the RNA molecules that have already been transcribed. Okay, after you identify these variations, there's many things that you really need to do as well. For example, you want to compare the disease sample against a control sample. You see, I identify a variation. I think it's important, but it's also in my control. That's not going to be important, right? So you have to do the controls. And also you can compare that against the database of common variants. And those are the variation. And actually, there's a database called the database of common variants. So you can go to look into that. And the notion is if it's a common, and those are the variants that doesn't reportedly causing any phenotypical changes. And you want to get rid of those as well. And then after going through this, and you probably, hopefully, you will get your candidate genes. And any people who worked on this data set will know it's not as simple as this at all, right? But it's still, that's the general way to go. So you identify the variants, you look for the control samples or common variants, get rid of those, and further derive which gene is causing the disease. Okay, and the scope of the resequencing, so whole genome sequencing is too expensive. So therefore, you can do the target sequencing. And so there's several ways that you can do the targeting, selecting the targets. The first one is to do the exon sequencing. So basically, we think that exons are most important part of the genome, right? Although they are not quite, but they are kind of become a protein if the nucleotide on the exons are changed. It can cause the protein changes, therefore cause bad things to happen. So that is really the exon sequencing has become very popular. And you can also do some other type of targeting, including, you know that I have a gene that I have studied my whole life on, and I want to deep sequence in the entire region, and that is possible as well. And we know that so you can really do the exons or whole genes, right? And this is one of the examples from our collaborators. So for one disease, they found strong indication from earlier studies that part of chromosome 2 is a problem for this disease. So they can really design the target assays to target only these regions to further deep sequence in those regions. So we will have more detail when we get into the DNA sequencing lectures. The technology to do this can be a capture array or capturing solution or any other PCR-based assay. And another interesting application is you think, I don't have money, I really want to do the sequencing, and you can still do by using 2 different samples into it. And the goal is to identify genetic variations with a cohort of samples. So in the third lecture, and we will very systematically talk about all these different DNA sequencing applications. There's several studies published. I think all of them are from last year, or this year. And the first one is Sick-Senos syndrome. And they did a whole genome sequencing and identified the genes that will cause this disease. And the Miller syndrome, this one is another one. They did exon sequencing. And this is for the human complex 1 deficiency. And they did a pooled sequencing and to help us to identify the genes. So there are several very successful applications on the DNA sequencing part. And apparently they're all published in Nature Genetics. And this DNA sequencing, because of the throughput, can help us to also study the copy number variation. So if you think of human genome, we are diploid genome, right? And so each gene, you should have 2 copies. But it's not always like that. Some part of the genome, they tend to have multiple copies, and especially in cancer, and those can really cause a big problem. But a shocking thing published in Nature 2006 is analysis of hypermap collections. Hypermap are some individuals, a collection of individuals they don't have strong phenotypes. They don't have disease. Okay, they're healthy individuals, and they found that CNV is in 12% of the genome contains copy number variation. That's the normal ones. Okay, and many are related to disease processes, but a lot of them are not. And the technology to study copy number variation has been mature before the sequencing even. And there's a microarray technology. You can really study the copy number variation, including Affymetrix arrays and Agilent and Nimbogen arrays. And also this array CGH, I'm not going to go into it, and you can also use that to study that. And this is one example to study copy number variation. This is using Agilent assays and Affymetrix assays, right? And you can study those, but the issue is the resolution. Okay, with this type of array-based technologies, sometimes the resolution cannot be very high. Okay, and but using sequencing, you can really use it with much higher resolution and no practical limit if sequence is deep enough. Okay. So copy number variation has been a big issue recently, and we see that there's a recent publication from Nature, and they did the copy number variation analysis from the 1,000 genome data, and we can go through that once we get to there. Copy number variation is one type of structure variation. Okay, there's many other type of structure variation as well. So the genome contains many structure variations, including the insertions, deletions, inversions, tandem duplicates, translocations, and a lot of other type of rearrangement. We don't even know the name, right? There's no name for that. It's just too complicated. And study these structure variations is really a big problem, because since we don't know where they are, and there's no way we can design arrays to target on those, so it wasn't really possible. But for very large ones, you can detect with FISH. The FISH is fluorescence in situ hybridization, and we know that for the genetic testing, if you do the Down syndrome, I think you've got 3 copies of chromosome 21, and Adler syndrome, 3 copies for chromosome 18. So those large structure variations, you can identify those using this FISH technology. But most of the variations we really don't know. We don't have a clue. And because of this sequencing technology, this becomes a really hot research topic that has been brought into a lot of disease mechanisms identification. And most of these are really hard to detect by the arrays. And mate pair libraries, we will talk about that a little bit, with different lengths of insertion sites can be informative. So what that really means. The so-called mate pair or pair-end technology, remember that we got a sequence, and we can sequence one end, but we can also sequence both 2 ends, right? And this is called either mate pair or pair ends. There are subtle differences. But the point is, for one sequence DNA fragment, you can sequence both 2 ends, right? And if this is the people you're studying, and you know that between these 2 ends, all my DNA fragment is about 300 base pairs. Just give a number, okay? 300 base pairs. However, when you align it back to the reference genome, you found that these 2 are 10,000 base pair away from each other. So there's something going on there, right? So a possible cause of that is this is the reference genome, and this is the ones that you're sequencing, and there's a big chunk has been deleted from the sequence that you are interested in. So this is one very good way to identify the deletions. And similarly, if you think this is my sequence race, it should be 5,000 base pair, but coming back to here, I only 300 left, and that means there's an insertion in your sequence rather than deletion. And I'm not going to go through all these complicated patterns. It can be inversion, so you can see that the direction has been different, and duplication, translocation, and this translocation from a different chromosome along insertions. So you can go through those and take a look at. This is a really cool study. I think it represents one of the futures of the future translational science. So, which is using this technology to study the patient-specific biomarkers. We are very familiar with biomarkers, right? We are trying to identify those. You got the tumor samples and do the microarray. We want to see which gene really help us to distinguish which drug or treatment is useful, things like that, right? And but patient-specific biomarkers, by definition, every single people have their own biomarker. So what this study tells us is, you can go back to it, it's a really interesting study. So you see in this is the cancer sample, tumor sample, and there's a big genome rearrangement here. So this red one doesn't supposed to be there, okay? And because of this cancer, so it's coming fused here for some reason. And then when you do the next generation sequencing, do the mate pairing, you found all those blues are pretty good, but there are two pairs. You see one part is blue, the other is red, and this has been replicated here, okay? And this really represents this patient-specific rearrangement that you really want to get resolved. And at that point, if you do the treatment for the patient, and you do different therapies, and you can really only target these small parts. We already identified your sequencing, and to see how well that this therapy is effective for this cancer. Okay, so I think this is a very important study that we should all pay attention to. RNA sequencing, okay, by definition, is to sequence the RNA. Should we make another 5 minutes break? Everybody stay put. Is that too long? We still have about half an hour lecture to go. All right, what did you say? Okay, keep going. All right, he's the one to blame. Okay, if you feel tired and you want that break. All right. So this is the RNA sequencing platform. So you can see that this is RNA and it's the poly A by the end of it. And here you want to sequence the RNA, and you need to do the RNA fragmentation and then break it into small pieces, put the linkers on, and then eventually you just sequence these individual RNA sequence molecules. And then after you get all these sequence read out, and you want to do the realignment, align it back to the human genome, so you will see that some of the reads really clustered around this exon region. This we call the exon reads, and some of them goes to the junction reads, and the others goes to the poly A read, polyadenylation part. So these are the ways that help you to sequence the transcriptome, and which is can be very useful. And the goal of the RNA sequencing technology is sequence the transcriptome to measure the complete set of transcripts in a cell and their quantity. So which part of the genome has been expressed and by how much. That's our goal. The advantage here is compared to arrays is this is a digital readout. So basically, for each individual genes, you want to count the numbers of reads that has been sequenced. And comparing to array technology, this is really higher dynamic range. And you can also find the novel transcripts, including the splice variants, different 5' 3' ends, and mutations and recombinations. All these things, it's impossible to be found on microarray technology because you don't know they exist there. And you can also find other genetic variations on these transcripts. And study allele-specific expression if your coverage is very deep. Okay, I'm going to skip this slide. I know it's very complicated, and the reason is we will have 3 lectures to talk about RNA sequencing. But the point I want to give you today is RNA sequencing is sequence the RNA. All right, that's pretty simple. And the next category of application is called the ChIP-seq, which stands for chromatin immunoprecipitation following next-generation sequencing. The goal here is to identify genome-wide binding patterns of a protein of interest. That protein can be a transcription factor, can be a histone-modification markers with different markers. So what happens here is you got a double-stranded DNA, okay, and you got a protein binds on there. So the point of this assay is you want to know that which part of the genome this protein targets to. The way to do that is you will first cross-link this double-strand DNA with the protein, okay? And then you break those into small pieces. And then you use antibody to target your protein of interest. So only those proteins that initially with those DNA fragments will be, put down here, and others will be washed away, okay? And then you only have this red double-strand DNA left, and then you go ahead to do the sequencing, and that will help you to identify which part of the genome this protein originally binds to, okay? So we will have a couple of lectures on that as well. This is exactly the same figure, and which is just prettier. So you can see the point here is this is the histone markers and the histone modification marker, and there's a transcription factor binding here. So with ChIP-seq, you can do both the transcription factor and the histone markers. It depends on the protein you want to, the antibody you use to target which protein to target. So this is a very nice review paper. We'll go through extensively when we get there. So a couple for ChIP-seq studies published with pretty significant impact, and actually ChIP-seq is one of the first applications of next-generation sequencing, and most publications I'm introducing here are published in 2007. And this is the first ever ChIP-seq data was published from Science in 2007. And they later identified the binding sites of the protein they're interested in, okay? And this is the same year and published in Cell. Basically here, they used antibody to target histone markers, not the transcription factors. And those are very interesting studies as well. And this is another one published in 2007, and the target on a transcription factor called STAT1. So they identified, I think, over 30,000 STAT1 binding sites in the human genome. Okay. A little tired, and so we go through this quicker. So next generation sequencing enables breakthrough in genetic studies. So in the next several slides, I'm going to give you a brief overview of several studies that are not able, there's impossible to do without this technology. So those are all we call the big science, and I never know what that really means, but let's go through this kind of examples. The first one is the Southern Genome Project. So it's not really individual lab can do it. It's consortium and many labs, and the NIH put big monies in there. And the goal here is to provide a deep categorization of human genome sequence variations. Okay, so we are not happy with only the reference genome anymore. We want to know what is the variation in our human genome. And there was 3 pilot studies published, projects here. And the first one is a low coverage of whole genome sequencing of 179 individuals. And it's only very low coverage. They cover the human genome only by 4 times, 4x coverage, very low coverage. And they also did a very high coverage sequencing of 2 trials, mother-father-child trials, very high coverage. And they also did a very good exon target sequencing of almost 700 individuals from 7 different populations. And these are the 2 major papers published out of Southern Genome Project, both from Nature. And so what we learned from Southern Genome Project, okay, and they identified locations allele frequencies and the local haplotypes for 15 million SNPs. And we found the 15 million SNPs in human genome. And 1 million short insertion and deletions. I'm talking about here are healthy individuals, okay, no disease. And 20,000 structure variations in some of the individuals. And each person carries about less than 300 loss of function variations in annotated genes and 50 to 100 variants in implicated inherited disorders, although those are healthy individuals. So the point of this is, of course, you can go back to the papers to figure this out, but the point is we are not happy with the reference genome anymore. We want to know how different our individuals are. We already know how different we are from monkeys, right? The sequence has been done for different species, but we want to know how different we are among us, and those can help us to identify the disease variations in the future studies. Another big consortium study is called the Cancer Genome Atlas. It's TCGA project. And they had a pilot phase, which is, this is really study cancer genome. And the pilot phase in the past 4 years, and they already published in glioblastoma and ovarian cancer this year, and the lung cancer is also in the pilot phase. And the platforms that they used in this TCGA project, you can see this is initially it's all array technologies, because it was 4 years ago, sequencing was still very expensive, right? And they already selected the best technology at the moment was available to target all these things. So you can see they will look for the array CGH, which apparently is a copy number variation. And this is the Affymetrix platform to study gene expression, to do the genotyping, microRNA, DNA methylation, alternative splicing, and other targeted sequencing. So they're really trying to understand the genome, pick out one cancer type, and from all different domains to study those. This is clearly a consortium type of work. And again, they didn't pick the sequencing, and they thought they can do it faster, but it turns out to be that the most hurdle were not from the technology or even analysis. The biggest problem is they had a hard time to get high quality samples and to really do this type of experiment. And initially they did some survey, and in this hospital they have how many samples, in that hospital they have how many samples. And they are good quality samples, but it turns out to be only about 15% of samples are usable in some of these studies. This really hold them up quite a bit. Maybe holding up is not a bad thing, because the technology becomes much more powerful 4 years from the pilot started. The main phase here is $275 million to provide more than 20 types of cancers and 500 per tumor types in the next 5 years, and in the next 1.5 years, and 2,000 pairs from 10 tumor types. So we really look forward to see the data availability. And for these studies, the data, once they are generated, they will be put into the public domain. That's the good news. The bad news is you won't get it. Okay, so they will be put there, and because the genome has a lot of information that can uniquely identify individuals, and those are really a lot of ethical issues needs to be figured out. So what they do is they still allow you to do the data, to use the data, but you have to write a proposal to the consortium, and they have to evaluate that, and they have to approve that. And then once, anytime that you want to increase a bit more, you need to write another proposal. So it's kind of holding up the process, but data can be available if you are really aggressive. And these are the sites of this Cancer Genome Project sites, okay? And similarly, there's another international Cancer Genome Consortium effort. Just, I think last year we have, what's the person named? Tim Hudson, and came here to give a talk. And he is that right, Ken? Yeah, he is he is the PI, one of the organizers of this International Cancer Genome Consortium. And it's so many different countries were involved and kind of formed the internal competition as well. All right, so Southern Genome Project, the Cancer Genome Project, all of those projects are because we're not happy with the reference genome anymore. We want to know a little bit more about it and to know the variations. And Southern Genome Project are like that. And there's another domain of information that we are not really familiar with yet. So once you got the 3 billion letters human genome sequence, not all of the 3 billion letters are useful. Some of the parts are more useful than others. We know the gene, the exon regions represent about less than 2% of the human genome, and a lot of people really spend effort on that. However, how to really annotate the meaning of those 3 billion letters is a big, big effort. And that comes with the ENCODE project, which is the encyclopedia of DNA elements. And this project started from initially launched in 2003. And at that time, there's just no other way. They want to do the whole genome. This is just not possible because of the technology limitation back in 2003. And they interrogate 1% of the genome, which is 13 million base. Half of them are manually selected, half of them are randomly selected. And the first paper they published from ENCODE project is 2007. And they identified quite a few things that if you're interested, you can go back to the paper. And there's 2 follow-up projects, and now becomes available, which is a full-scale human study, not only the 1% of genome, but the full scale, and also some of the model organisms. And the most notably is the Drosophila and C. elegans. Okay, Drosophila is the flies, and C. elegans are the worms. And those are really interesting the models that people study biology. And for the ENCODE project, initially they are all array-based technologies. You can see this RNA transcripts, promoter enhancers, DNA methylation, chromatin modification, transcription factor binding, DNase-1 hypersensitivity, and regions of replications. So all the array-based technology. But now the newer phase, of course, it has to be sequencing-based technology. And this is the model organism part, which is called the mode ENCODE. And basically, it's Drosophila and C. elegans. And so we already have the paper in the ENCORE system if you're interested in looking at. So you can see this is based on these 2 model organisms. You can look for transcription, splicing, which part of the genome has been transcribed, and the replications, epigenetics, including histones and methylations, and transcription regulations, and which part is the enhancer, promoter, and all different type of interesting stuff. And you can go back to the paper to check the availability. Okay, so there are some big science is available and based on this technology. But I want to make sure that this technology is also available for smaller labs. So if you study individual one disease samples, you want to do the transcription analysis, it's very much possible and it becomes more and more affordable. And I didn't talk about it here. That doesn't mean they are not there. So you can really still use that in your laboratory. The next part of this talk is the basic concepts. There's 2 basic concepts I want to deliver, and we will talk about that repeatedly in the later lectures. The first one is pair ends and mate pairs. Okay, so we talk about it initially. So we got a DNA sequence, which is different lengths of DNA sequences, and this is a single end. So you can see we will sequence the very beginning part of the DNA. Okay, and pair end is a different concept. So you can see that the fragment length is about 200 and 500 base pairs, still that much. And instead of sequencing one end, you sequence both 2 ends. Okay, for 2 different platforms, they are different. So you can see this is 100 base pair each, and this is 75 versus 35 base pair each. Okay? And this is the pair end. And what it's good for is good for RNA sequencing technology. And also it increased alignment accuracy because we know human genome are kind of very repetitive. So sometimes you got 100 base pairs. You hope that you can uniquely map to the human genome, but because of the repetitiveness, you cannot do that. However, once you sequence another 100 base pair with 500 base pair away, and it's unlikely that part is also in the repetitive regions, so it can help you to anchor the alignment. That really increases the alignment accuracy. And similarly for the solid platform. And this comes to the confusing part. There's another thing called the mate pairs. Okay? And these are the concepts that you need to know. Okay, these are concepts you need to know. I know this is boring, but you need to know. So the difference between the pair ends and the mate pairs are the fragment lengths are different. For the pair end, for the mate pair end, it's short. For the mate pair, it can be 2 to 5 kilobase away from each other. Okay, and another difference here is the direction of the sequencing is different. Okay, you can see the mate pairs is sequenced this way, and for the pair end, you sequence it inwards. Okay, for the solid platform, it's also longer, and the direction is different as well. So it's kind of can be confusing. It just depends on how they are doing their biochemistry. But the point here is this is something you really need to keep in your mind. When you heard about people talk about the pair end and the mate pairs, they are different. They are not the same thing. Okay, conceptually, you got a DNA molecule, you sequence both two ends. Okay, however, the length of the fragments are really different. Mate pairs are much longer. Okay, and actually their biochemistry behind that is different as well. I'm not going to get into. And this is really good for de novo assembly and good for identifying structure variations for the mate pairs. Another concept, and it's multiplexing, which is barcode. For some of the applications, we may not need to generate so much reads per sample. It wasn't an issue a couple of years ago when these sequencers, the throughput are not very high, but it is a problem right now. It's a good problem to solve, right? So for each lane of Illumina technology, you generate so much data, hundreds million reads. For some studies, I really don't need that many. So how I can use a small part of that entire read lane to study that? So what you can do is a multiplexing, which is pull samples into a single lane of a flow cell. Okay? And you can add a short index to the tag library. So basically you can see, think of there's one fragment, it's from sample number 1, I add up A here. For sample number 2, I add up a different letter in the beginning. And then after you do this, you mix them together and put them in the same lane. And then eventually when you do the bioinformatics, you see, oh, the first letter is A. This is from sample A. The first letter is T, it's from the second sample. So you can conceptually decipher that in the later stage. And for the Illumina technology and the multiplexing capacity, this is something you really need to consider when you design experiment. And of course, it's a moving target because they are both evolving. And for the Illumina, they use 6 base oligos to do the index multiplexing. And currently, there's 12 unique tags to generate 96 samples. So their multiplexing capacity is 12. So they can barcode 12 samples into one experimental units. For the solid, it's similar. It's up to 16 barcodes. And I think they just published a 96 barcode protocol, but still needs to be tested. Okay? So sometimes you can barcode 96 samples in one experiment. Okay. The challenge is, okay, the good thing of this part is that you don't have to use your brain. Okay, this is just a general information type. The bioinformatics is the real bottleneck. So when at a time the sequencing becomes so cheap, and bioinformatics, when a lot more data has been generated, and we are the problem, the bioinformatics. So researchers are able to generate the data faster than and more easily than their ability to analyze them. That's for sure. What is going on? Or going to analyze, who is going to analyze the data? That's a big issue. So for some big labs, when they generate a lot of data, they can hire their dedicated person to do the sequencing analysis. But for many smaller labs, that's not an option. So what is going to be the business model about sequencing analysis in real academic environment? That's a big thing that needs to be worried about. For some of the analysis, a large amount of processing power are required. Okay, it's really hard for you to do something on your desktop anymore, right? We have very powerful machines. Sometimes it's not, clusters. Sometimes it's still not good enough. And so I quit developing web-based software, okay? So we had experience, we developed some web-based software, and people submit their jobs, it totally blocked my server, so I cannot do anything with it anymore. So it's very, very difficult for the web-based studies, which are available for a lot of other applications that you might be familiar with. Software package might be useful in formulating hypotheses and carry out initial analysis. So people always think analysis can be simple, right? You just plug in that software and run it. Okay, that is good for the initial phase. However, manual intervention is necessary to tease out the noises and get to the publication stage. So there's a lot of efforts needs to be built into that part of it. I think Janice knows that perfectly, right? And Janice, by the way, is working with Tatiana to struggle on those data analysis. And we know those, and we need time to spend on those. And that's one of the reasons I think it's a good idea for biology students come here to this class and to know those our struggles and where potential noise come from. And then you can do the analysis in a more intelligent way. And some investigators can learn, but not everybody can learn. Sometimes it's hard. And the problem will become more acute in the future with more and more sequencing data. The last topic here is related to the challenge. We have so many data, and it's not conceivable that every lab you generate a set of sequencing data, you have to buy a big computer cluster. It's impossible, right? It's just like when you do a sequencing data experiment, you don't have to buy a sequencer, right? You send your samples to somewhere, and they will do that. The overhead, the maintenance for those sequencers, you don't have to deal with it. The same thing is for the computation part. Okay, we need big machines, but that doesn't mean you have to buy big machines. So cloud is a very interesting idea to distribute your computation to, into a much scalable scale. So you can see the cloud are a large pool of easily usable and accessible virtualized resource. And this resource can be dynamically reconfigured to adjust a variable load. You can read those things, but the point is you don't have to buy your computer. There are computers somewhere in the world, right? It doesn't matter where that is, right? Some part Google has it or Amazon has it. It doesn't really matter. So what you want to do is once you get your data analysis done, you send your samples to one of the computational resources somewhere in the world, and then they can help you to do it. They already help people to maintain those big machines, so you don't have, and then you just pay what type of computational time you used. Okay, so the point here is ability to scale. Okay, let's say jobs that takes 10 hours on single server can be done in 1 hour on 10 servers. Okay, so if you spend 10 hours, 1 hour on 10 servers, and the servers you can distribute the cost, and that really saves you a lot of your time. Pay per use, only pay for what you use when you use it. Okay, when you don't use it, you don't pay for it. Okay, some people, it's the electricity. I mean, for those big servers, it's not like your desktop. You just throw it home and it doesn't cost anything. For those big servers, you need a cooling system. You need a UPS to protect the surge of the power. It's very kind of intensive job to do that. And it also reduces the needs to purchase the hardwares. And increase the flexibility is a variety of server types and operation systems. So for different type of applications, you may use different node type of server. So let's get to this. So you can see that you can really use small nodes, okay? And these are not powerful machines, okay? This is Amazon Web Service pricing. And it's about 10 cents per hour for a core, like 1.7 gig of memory and 160 gig of storage. Not big at all, right? But you only pay 10 cents per hour when you use it. And there's larger ones that have larger memory and storage and more computational power, and you pay a little bit more. And there's some bigger ones as well, okay? So the point here is when you do different type of study, after you get your experiment done, some of the applications you may want to use the smaller ones, others you may want to use the bigger ones, and you don't have to buy two machines. You just use their service, and it's very much scalable. However, if you don't use the cloud very often, and sometimes you will make a mistake, you can spend thousands of dollars and don't get anything out of it, right? And so you analyze the details of the projects to take full advantage of the cloud. So that's something you have to do. So create a backup strategy before you launch an instance, okay? So don't just trust the cloud service. They will maintain your data very well. You have to back up your own stuff. And only launch an instance when you're ready to start working and shut down the instance when the work is complete. The reason for that is the charging, the timing starts from the time you kick in, right? Not start from you run the real data. So if you don't plan well, some of the servers, sometimes we know that one job is dependent on the completion of another job and things like that. If you don't do it well, and a lot of servers you're holding there, you're paying for it, and not really using it. And when you get to use it, and a lot of money has been wasted. So just really a lot of things needs to be planned out before you use it. And access an instance using a secure connection is important, and you have to actively monitor your account. Otherwise, your credit card will be going away pretty fast. And actually, in addition to this, there's another thing that needs to be considered, which is the IRB issue. Because we are dealing with the genetic material, and I mean, there's so much data there. We kind of trust Amazon. They are pretty secure. Nobody hack into it or things like that. Well, however, can you convince the IRB on that? If for anything, they are leaking information, and someone will be involved into the lawsuit, right? And they are not HIPAA compliant. And so for a lot of real patient data, if you didn't get approved, you probably cannot use cloud computing. That's the downside of it. But I assume that Amazon needs to figure that out by themselves. Once they become HIPAA compliant, we can use it. And another thing is a company like Eli Lilly, they have the policy, their data cannot be run on cloud. So we still need to buy bigger machines sometime. All right, this is what I'm going to cover today, and I know you're tired, but I'm tired. For the next lecture, okay, so we will basically talking about a lot of boring things, okay? A lot of things that for the students who registered, you really have to go through it. It's painful. It's going to be very painful, but it can be useful to a certain extent. For the data analysis workflow and sequencing quality evaluation, these are the error rate estimation, those things. And a very big chunk of my next lecture will be on the alignment algorithm. So I will give you a general overview of my understanding of alignment and how that has been completed. Another big part I want to talk about next time is the data formats. Okay? Boring thing, right? But you have to know those if you really want to do the analysis. So And also for the data visualization. So if for some of the people who just want to sit in the class and the next time, depends on your interest, you may or may not come here. But if you don't come next time, I encourage you to come for the third lecture, which is a DNA sequencing, a general biological application. I'm going to give you more introduction in that area. Okay? All right, so again, if you are sitting in the class and I know that you probably sent me email already, but still send an email to Hao Yu, and he will create an account for you in the Encore system so you can access all the materials, including the slides. All right? So I'll see you next week.